Hello everyone, my name's Ima and welcome back to my YouTube channel. Today we're going to cover chapter 7 for our MCAT biology playlist, and this chapter is titled The Cardiovascular System. In this chapter, we're going to cover the following objectives. First, we're going to talk about the anatomy of the cardiovascular system. That includes the heart itself, the vessels that carry the blood throughout the body, known as the vasculature, and the overall circulation pathway that connects everything together. Then we're going to take a closer look at blood. We're going to break down its composition, meaning what's actually in blood. And we're also going to talk about blood antigens, which are really important for understanding blood types and immune reactions. Finally, we're going to cover the physiology of the cardiovascular system. That means we're going to look at how this system actually works in practice, and we're going to cover things like blood pressure, gas and solute exchange between blood and tissues, and the process of coagulation or blood clotting. With this introduction in place, let's go ahead and get started with Objective 1, the anatomy of the cardiovascular system. The cardiovascular system is composed of three major components: the heart, the blood vessels, and blood. The heart is a muscular organ with 4 chambers, and it functions as a pump. Its job is to keep blood moving through a network of blood vessels that we refer to as vasculature. This vasculature includes 3 main types: arteries, capillaries, and veins. First, let's get a broad overview of how blood moves through the heart, lungs, and body. After circulating through the body, deoxygenated blood is going to return to the heart through the veins. It enters the right side of the heart, where it's pumped to the lungs to release carbon dioxide and pick up oxygen. The oxygenated blood then returns to the left side of the heart, where it's pumped out to the rest of the body. And this continuous cycle is how oxygen and nutrients are delivered to tissues, while waste products are collected for removal. Now, if you look at the diagram on the screen, you'll see the labeled structure of the heart. The heart has 4 chambers, and it functions as 2 pumps working in series. The right side of the heart pumps deoxygenated blood to the lungs. This is called pulmonary circulation. And the left side of the heart pumps oxygenated blood to the body. This is known as systemic circulation. Each side of the heart has 2 chambers: an atrium, which receives blood, and a ventricle, which pumps blood out. So in total, we have the right atrium and right ventricle, and the left atrium and left ventricle. Now let's break it down step by step and follow the exact path that blood takes as it flows through the chambers of the heart. Blood from the body enters the right atrium through 2 large veins, the superior vena cava and the inferior vena cava. From the right atrium, it flows into the right ventricle, which pumps it through the pulmonary artery to the lungs. In the lungs, gas exchange occurs, so carbon dioxide is released and oxygen is absorbed. The oxygenated blood then returns to the heart via the pulmonary veins, and it enters the left atrium. It moves from the left atrium into the left ventricle, which pumps it out through the aorta. The aorta is the largest artery in the body, and it does that so it can deliver oxygen-rich blood to all tissues. The heart contracts in a specific sequence. First, the atria contract. That is going to push blood into the ventricles. Then the ventricles contract to send blood either to the lungs or to the body, depending on the side of the heart. And this kind of coordination is very important to maintain continuous and efficient blood flow. But there's something else that keeps everything on track, and that is the valves. Valves are critical for preventing backflow and making sure that blood moves in only one direction. Without valves, the high pressure that's generated during contraction could push blood in the wrong direction. With that being said, there are 2 main types of valves in the heart. You have the atrioventricular valves, AV valves, and the semilunar valves. The AV valves, they're located between the atria and the ventricles. So on the right side, we have the tricuspid valve that separates the right atrium from the right ventricle. On the left side, we have the bicuspid valve, and this separates the left atrium from the left ventricle. As for the semilunar valves, they're located at the exits of the ventricles. So the pulmonary valve, it sits between the right ventricle and the pulmonary artery, and the aortic valve is between the left ventricle and the aorta. These valves, they open and they close in response to pressure differences created during each heartbeat. They make sure that blood moves forward through the heart and out into circulation. Now, one final structural point I want to make. The left ventricle, it has a much thicker muscular wall than the right. And this is because it has to generate enough force to send blood throughout the entire body, whereas the right ventricle only needs to push blood to the lungs, which are nearby, and they're under much lower resistance. With this overview of the heart structure and blood flow in place, now we're ready to look at how the heart knows when to contract, specifically how electrical signals coordinate the timing of each beat. The heart, it doesn't rely on nerves to tell it when to beat. Instead, it has its own internal conduction system that's made of specialized muscle cells that will generate and transmit electrical signals. These signals control the timing and they control the coordination of each heartbeat. So they make sure that the atria contract first, and then that's followed by the ventricles. So let's go through the pathway of electrical excitation step by step. It all starts at the sinoatrial node, also known as the SA node, and it's located in the wall of the right atrium. This is the natural pacemaker of the heart. Now, the SA node, it generates regular electrical impulses, about 60 to 100 signals per minute. And that sets the rhythm of the heart. That's why the normal resting heart rate is somewhere between 60 to 100 beats per minute. From the SA node, the electrical signal is going to spread through the atria, and it causes them to contract and push blood into the ventricles. The signal then reaches the atrioventricular node, or AV node. And this is located between the atria and the ventricles. The AV node, it acts like a gatekeeper. So it'll briefly delay the signal to allow the ventricles time to fill completely before they contract. After the AV node, the signal travels down the bundle of His, which is a pathway of fibers that runs down the interventricular septum. That is the wall between the two ventricles. This bundle then branches into the left and right bundle branches, which carry the impulse down either side of the septum. Finally, the signal reaches the Purkinje fibers. They spread throughout the walls of both ventricles. And these fibers, they will rapidly distribute the electrical impulse to the ventricular muscle cells. And that will trigger a strong and coordinated contraction that pumps blood out of the heart. Now, this electrical conduction system is also what produces the waves that you see on this electrocardiogram, or ECG. So the P wave, it corresponds to atrial depolarization, or the contraction of the atria. The QRS complex represents ventricular depolarization when the ventricles contract, and the T wave shows ventricular repolarization as the ventricles reset for the next cycle. Now that we understand this, let's take a moment to learn what's actually happening during one full heartbeat. This is called the cardiac cycle, and it consists of two main phases, systole and diastole. Systole refers to the phase when the ventricles contract. So during this time, the atrioventricular valves, or AV valves, they're closed, and the semilunar valves are open. This is going to allow blood to be pumped out of the ventricles, either into the aorta or the pulmonary artery. Then comes diastole, which is the phase when the heart relaxes. So during this, the semilunar valves are closed and the AV valves are open. This allows the ventricles to fill with blood again, which prepares it for the next contraction. If you look at this diagram, you'll notice it brings together several measurements, changes in pressure, volume, and electrical activity, and they're all mapped over the course of the cardiac cycle. You're not expected to memorize every detail here for the MCAT, but it's helpful to recognize how everything is interconnected. So for example, when the ventricles contract during systole, ventricular pressure increases, volume decreases, and blood is pushed out. You'll also notice that the first heart sound happens when the AV valves close. Then during diastole, ventricular pressure drops, volume increases, and the second heart sound occurs as the semilunar valves close. These phases are going to repeat with every heartbeat, and they drive the circulation of blood throughout the body. Now let's tie this into an important concept called cardiac output. Cardiac output is the total volume of blood that's pumped by one ventricle in 1 minute. And it depends on two things: the heart rate, which is how many beats per minute, and the stroke volume, which is how much blood is pumped out with each beat. So the formula is cardiac output equals heart rate multiplied by stroke volume. For adults at rest, cardiac output is around 5 liters per minute, but this number isn't fixed. For example, it increases during exercise when the heart beats faster and it pumps more blood with each contraction. And it can decrease during rest or even in certain medical conditions. Then, to distribute blood efficiently throughout the body, the cardiovascular system relies on a branching network of vessels. This system is known as the vasculature, and it's made of arteries, veins, and capillaries, each with a different structure and function. And the design of this system is incredibly important because blood must reach every tissue from the brain down to the toes. And to do that, blood flows through vessels of progressively changing size. So let's walk through the general flow. Blood leaves the heart through large arteries. These branch into smaller arterioles, which lead into the tiny capillaries, which is the site of exchange. After passing through the capillary beds, blood moves into small venules, which merge into larger veins that carry blood back to the heart. Now, one thing all these blood vessels have in common is that they are lined with endothelial cells. These cells form a thin, smooth layer on the inner surface of vessels, and they help maintain vessel integrity, and they help regulate blood flow, and they allow substances to pass in and out of the bloodstream. Now, let's really break down the 3 main types of vessels, starting with arteries. Arteries carry blood away from the heart, and in most cases, that blood is oxygenated. The exception is the pulmonary arteries, which carry deoxygenated blood to the lungs. Now, arteries, they have thick muscular walls that can withstand and maintain high pressure, and their elasticity helps absorb the force of each heartbeat and then helps maintain blood pressure during relaxation. As arteries branch out and become smaller, they form arterioles. Arterioles still have muscular walls, but their main role is to regulate blood flow and pressure by changing their diameter. They essentially create resistance in the system, and that's important for directing blood to the tissues that need it the most. Next, we reach the capillaries, which are the smallest and thinnest blood vessels in the body. Actually, their walls are just 1 cell thick, and they're made of a single layer of endothelial cells. That kind of structure really allows for efficient exchange of gases, nutrients, and waste between the blood and the surrounding tissues. Blood flow here is going to be slow, and that gives time for substances to diffuse. But capillaries are also fragile, and if they rupture, that can lead to bruising or bleeding. After blood passes through the capillaries, it enters small venules, and from there it moves into larger veins. Veins carry blood toward the heart, and most of the time, this blood is deoxygenated, except for pulmonary and umbilical veins. Compared to arteries, veins are going to have thinner walls and less smooth muscle. They operate under much lower pressure, so they're going to rely on valves to prevent blood from flowing backward, especially in the limbs where blood has to move upward against gravity. In addition to valves, skeletal muscle contractions really helps push blood through veins, particularly in the legs. This is why long periods of inactivity, like during a long flight or bed rest, that can increase the risk of deep vein thrombosis or DVT. A clot can form, and if it travels to the lungs, that can cause a dangerous pulmonary embolism. Now, if you look at this image, you're going to see the structural differences between arteries, veins, and capillaries. Arteries have the thickest walls with multiple layers. Veins have similar layers, but they're thinner. And capillaries, on the other hand, they're just made of a basement membrane and endothelium, nothing more. Finally, to bring everything together, let's look at the complete circulation pathway, how blood moves through the heart, lungs, and body in one full cycle. We're going to start at the right atrium, where blood returning from the body enters the heart through the superior and inferior vena cava. From there, blood passes through the tricuspid valve into the right ventricle. When the right ventricle contracts, blood is pushed through the pulmonary valve into the pulmonary artery, which carries it to the lungs. This is where gas exchange happens. So carbon dioxide is released and oxygen is absorbed. The now oxygenated blood returns to the heart through the pulmonary veins, entering the left atrium. It then flows through the bicuspid valve into the left ventricle. With the next contraction, the blood is pumped through the aortic valve into the aorta, which sends it into systemic circulation. From the aorta, blood flows into large arteries, then into smaller arterioles, and eventually into the capillaries, where nutrient and gas exchange occurs with the tissues. From there, blood moves into venules, then into veins, and it finally returns back to the right atrium through the vena cava, where the cycle begins again. And this pattern, heart to lungs, lungs to heart to body, and body back to heart, it's continuous, and it's very essential for our survival. Now, in most cases, blood only passes through one capillary bed before returning to the heart. But there are a few important exceptions known as portal systems, where blood flows through two capillary beds in series before completing the loop. There are three portal systems in the body. The first is the hepatic portal system, which routes blood from the digestive tract to the liver before returning it to the heart. Then we have the hypophyseal portal system, which carries hormones from the hypothalamus to the anterior pituitary. And finally, we have the renal portal system, which routes blood through the glomerulus and then another capillary network in the kidneys. And these systems, they allow for extra processing, regulation, or exchange at specific sites in the body before the blood rejoins the general circulation. We have now completed Objective 1, and we're ready to move into Objective 2. Objective 2 focuses on blood. Now, by volume, blood is about 55% liquid and 45% cells. The liquid portion is called plasma, and the cellular portion includes red blood cells, white blood cells, and platelets. Let's start with discussing the plasma. Plasma is a pale yellow fluid that makes up just over half of our blood volume. It's mostly water, but it also contains proteins, hormones, nutrients, waste products, and even clotting factors. The three major plasma proteins are albumin, which helps maintain osmotic pressure, globulins, which include antibodies, and fibrinogen, which is important for clot formation. Now let's look at the cellular components of blood, starting with erythrocytes, also known as red blood cells. Red blood cells have one main job. That job is to transport oxygen from the lungs to the tissues and then remove carbon dioxide and waste from the tissues back to the lungs. And to do this efficiently, red blood cells are packed with hemoglobin, which is a protein that binds oxygen. Each hemoglobin molecule can carry up to 4 oxygen molecules at a time. To maximize space for hemoglobin, mature red blood cells lack a nucleus and other organelles. They also have a very specific shape. That shape is called a biconcave disc. This shape increases surface area for gas exchange, and it gives the cells flexibility so that they can squeeze through tiny capillaries. Red blood cells are going to have a lifespan of about 120 days. After that, they're going to be removed and broken down by the spleen or liver. You may also hear the term hematocrit. This refers to the percentage of blood volume that's made up by red blood cells, and it's often used as a clinical measure of oxygen carrying capacity. Next, let's discuss leukocytes or white blood cells. White blood cells make up less than 1% of total blood volume, but they're so essential for immune defense. And there are 2 main categories of white blood cells. You have granulocytes and agranulocytes, and that's based on whether or not they contain visible granules in their cytoplasm. Let's start with granulocytes, which include neutrophils. These are the most abundant type. They are the first responders to infection, and they specialize in fighting bacteria. Then we have eosinophils. These cells target parasites, and they also play a role in mediating allergic responses. Finally, there's basophils. These are the least common type. They release histamine, and they contribute to inflammatory reactions, especially during allergies. Then we have the other category, agranulocytes, which do not have visible granules. Here we want to discuss lymphocytes and monocytes. Lymphocytes include B cells, T cells, and natural killer cells, and they play a key role in targeting specific pathogens. They're also responsible for long-term immunity. Monocytes, these circulate in the blood, and once they migrate into tissues, they become macrophages. Macrophages digest pathogens, they clear debris, and they're very important for chronic inflammation and immune signaling. So even though white blood cells are few in number, they provide diverse and powerful immune responses against infections and other threats. Finally, we can discuss thrombocytes or platelets. Platelets are not full cells. They're actually cell fragments that come from large bone marrow cells called megakaryocytes. Their main role is to initiate blood clotting. When a blood vessel is injured, platelets are going to stick to the exposed collagen, then they'll become activated, and they will release chemicals that recruit even more platelets to the site. And this essentially forms a temporary plug, and it begins the process of coagulation. Platelet production is regulated by a hormone called thrombopoietin, which is made primarily in the liver and kidneys. Now, if you look at the figure on this slide, you'll see an overview of hematopoiesis, which is the process by which all blood cells are formed from stem cells in the bone marrow. Now, here in this chapter, we're not going to discuss all the branches. We don't need to memorize it. This is going to be the focus of the next chapter, but I want you to be aware that erythrocytes, leukocytes, and platelets, they all originate from common stem cells. And again, we're going to come back to this in the next chapter when we talk more deeply about blood cell development. Now that we've discussed the components of blood, let's take a closer look at one of the most clinically important features of red blood cells, and that is their surface antigens. These are specific proteins or molecules that are found on the surface of red blood cells that the immune system can recognize. In general, an antigen is any substance that can trigger an immune response. And when it comes to blood types, there are 2 major antigen systems that we're going to discuss that we need to know for the MCAT, the ABO system and the Rh factor. Let's start with the ABO system. A person's ABO blood type is determined by which antigens are present on their red blood cells. If your red blood cells have A antigens, then you're type A. If they have B antigens, you're type B. If you have both, you're type AB, and if you have neither, you're type O. But in addition to the antigens on your red blood cells, your immune system also produces antibodies against the antigens you don't have. So someone with type A blood is going to have anti-B antibodies. Someone with type B blood is going to have anti-A antibodies. Type AB blood, it has no antibodies. And type O blood has both anti-A and anti-B antibodies. Now, in practice, we don't just classify people as type A, B, AB, or O. We also include a positive or negative designation, which refers to a second antigen called the Rh factor. The Rh factor is another protein that's found on the surface of red blood cells. The most important version is the D antigen. So if your red blood cells have the D antigen, then you're Rh positive. And if you don't, you're Rh negative. So for example, someone with A antigens and the Rh factor, they're going to be classified as A positive. Someone with B antigens but no Rh factor, they're going to be B negative. And that makes 8 possible blood types: A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. Understanding these blood groups is important, especially when it comes to blood transfusions. When someone receives a transfusion, their immune system is going to attack any red blood cells that carry unfamiliar antigens. So if you give type A blood to a type B person, the anti-A antibodies in their plasma, they're going to attack the donor cells. That's why transfusion compatibility is based on both ABO and Rh status. So then it becomes important to note that O negative is considered the universal donor for red blood cells because it has no A, B, or Rh antigens. And AB positive is the universal recipient because these individuals don't have any antibodies against A, B, or Rh antigens. Now, Rh status is extremely important in pregnancy. If an Rh negative mother carries an Rh positive fetus, fetal blood can sometimes mix with the mother's circulation during delivery. The mother's immune system, it may recognize the Rh antigen as a foreign, and then it's going to start making anti-Rh antibodies. Now, this usually doesn't affect the first pregnancy, but it does become a risk in future pregnancies if the fetus is again Rh positive. The mother's anti-Rh antibodies, they can cross the placenta and they can attack the red blood cells of the fetus, and that can lead to some complications. So to prevent this, Rh negative mothers are usually given an injection of RHOD immune globulins, which can block the immune response and protect future pregnancies. Now, as a little fun fact, while the ABO and the Rh systems are the most important and the ones that are the most emphasized on the MCAT and in most general courseware, there are actually other recognized blood group systems and hundreds of distinct blood types based on various combinations of surface antigens. If you guys are interested in this, let me know. Maybe I can link a couple of fun scientific articles to read about this. But all you need to know for the MCAT is the ABO and the Rh systems. And with that being said, we've officially completed Objective 2. Our last and final objective is titled Physiology of the Cardiovascular System. The cardiovascular system plays several key roles beyond just moving blood. It helps maintain blood pressure, it regulates gas and solute exchange, it enables clot formation when injuries occur, and it even contributes to thermoregulation. We're going to look at each of these functions one by one, starting with blood pressure. What exactly is blood pressure? Blood pressure is the force that blood exerts on the walls of blood vessels. This pressure is what keeps blood flowing through the circulatory system, and it's what allows oxygen and nutrients and hormones to reach tissues throughout the whole body. Without it, nothing would move. But if pressure gets too high or too low, that can then cause some serious problems. We measure blood pressure using a device that's commonly known as a blood pressure cuff. And blood pressure is going to be reported as two numbers. The first number is the systolic pressure, and this is the pressure in the arteries when the heart contracts. The second number is the diastolic pressure, and that is the pressure of when the heart relaxes between beats. So for example, a blood pressure of 120 over 80 means 120 millimeters of mercury during systole and 80 during diastole. Blood pressure is very tightly regulated by the body. One of the most important regulators is the smooth muscle in the walls of the arterioles, which are small branches of arteries. Arterioles control resistance, and they can constrict to increase resistance and raise pressure, or they can dilate to decrease resistance and lower pressure. That's why they're often referred to as resistance vessels. This is especially important in making sure that blood is directed to where it's needed the most, like the muscles during exercise, or to the digestive organs after a meal. If blood pressure is too high, that's called hypertension. Over time, high pressure can damage blood vessels and organs, especially the heart, the kidneys, the brain, and the eyes. If pressure is too low, then tissues won't get enough oxygen. The goal is to keep blood pressure just right. So Goldilocks blood pressure, not too high, not too low, so that blood moves forward effectively without causing harm. Now let's connect this concept to a key equation that describes how blood pressure is controlled. The equation is delta P equals CO, which is cardiac output, times TPR, which is total peripheral resistance. In other words, the pressure difference across the circulation equals how much blood the heart is pumping multiplied by how much resistance the vessels are providing. Now, if either cardiac output or resistance increases, then pressure goes up. If they decrease, then pressure goes down. Cardiac output depends on two things, if you remember from an earlier objective, that is heart rate and stroke volume. As for total peripheral resistance, that depends on how constricted or relaxed the blood vessels are, primarily the arterioles. Now, hormones also play a role in regulating pressure. For example, antidiuretic hormone increases water reabsorption in the kidneys, which increases blood volume and pressure. Aldosterone, that increases sodium reabsorption, which pulls water with it. ANP works in the opposite direction, so it helps lower blood pressure by promoting salt and water loss, and it causes vasodilation. Now, if you look at the graph here, you'll see how blood pressure changes as blood flows through the circulatory system. The highest pressure is in the aorta and the large arteries. Then as blood moves into the arterioles, there is a dramatic drop in pressure. This is where most of the resistance occurs. By the time that blood reaches the capillaries and then the veins, the pressure is much lower. And this pressure gradient is, again, what drives blood flow, and it's why blood can move from the heart to the toes and back again. Now, let's go ahead and discuss gas and solute exchange. This is another function of the circulatory system. Gas and solute exchange happens primarily in the capillaries, and it's driven by both blood pressure and concentration gradients. Blood pressure pushes blood forward into the narrow capillary beds, and this is where exchange takes place. At this point, oxygen and nutrients leave the blood and they enter the tissues, while waste products like carbon dioxide leave the tissues and move into the blood. Hormones can also diffuse in or out at this level depending on what the body needs at that moment. Again, diffusion always moves substances from high concentrations to low concentrations. That's why oxygen leaves the blood. It's at a higher concentration there, and it will enter the tissues where oxygen is lower. And the reverse is true for carbon dioxide. It builds up in tissues as a waste product, and it will move into the blood where its concentration is lower. With that being said, let's start by discussing oxygen in more detail first. Most of the oxygen in the blood, it's carried by hemoglobin, which is a protein found in red blood cells. Each hemoglobin molecule has 4 subunits, and each subunit contains a heme group that can bind 1 molecule of oxygen. So overall, 1 hemoglobin molecule can carry up to 4 oxygen molecules at once. Now, what's especially interesting is how oxygen binds to hemoglobin cooperatively. That means that once 1 oxygen molecule binds, hemoglobin changes shape slightly, and it makes it easier for the next oxygen to bind. So with each oxygen added, binding becomes easier. To understand how tightly hemoglobin holds onto oxygen, we have to look at something called the oxygen-hemoglobin dissociation curve. Along the x-axis, you will see the partial pressure of oxygen. On the y-axis is the percentage of hemoglobin saturated with oxygen. In the lungs, where oxygen pressure is high, hemoglobin is nearly fully saturated. It grabs onto oxygen tightly. But in the tissues, where oxygen pressure is lower, hemoglobin releases oxygen so that it can be used by cells. You can even see from the curve that during exercise, when tissues are using more oxygen, the oxygen pressure drops even lower, and that causes hemoglobin to release oxygen more readily. And that's perfect because those active tissues need a constant supply of oxygen. Now let's talk about what influences this oxygen release. Hemoglobin's affinity for oxygen, it isn't fixed. It responds to the environment around it. This is where we introduce the Bohr effect. The Bohr effect refers to the fact that increased levels of carbon dioxide and hydrogen ions, both of which are found in metabolically active tissues, they cause hemoglobin to release oxygen more readily. In other words, higher carbon dioxide or lower pH decreases hemoglobin's affinity for oxygen, and it makes it easier to offload oxygen where it's needed most. Let's break that down even further. Looking at this curve, a right shift of the curve means hemoglobin holds onto oxygen less tightly. It's more willing to let it go. This is favored by several factors: increased partial pressure of carbon dioxide, increased hydrogen ion concentration, which will lower the pH, increased temperature, and increased 2,3-DPG levels. 2,3-DPG is a molecule that's produced by red blood cells during glycolysis, and it binds to hemoglobin and it lowers its oxygen affinity. So when tissues are active and they're generating heat, carbon dioxide, and acid, all of these signals tell hemoglobin, hey, let go of the oxygen, we need it here. On the flip side, a left shift means hemoglobin holds on more tightly to oxygen. This happens when carbon dioxide levels are low, when pH is higher, so there's fewer hydrogen ions, when temperature is cooler, or when 2,3-DPG levels are lower. Also worth mentioning, fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Also, we don't want to forget about carbon dioxide's journey. Carbon dioxide is carried in the blood in 3 forms. First, there's a small amount that's dissolved directly in the plasma. Second, some carbon dioxide binds to hemoglobin, not at the oxygen binding sites, obviously. And third, and most importantly, carbon dioxide is converted to bicarbonate ions. This happens through the reaction of carbon dioxide with water, forming carbonic acid, which rapidly dissociates into hydrogen ions and bicarbonate. And this process is very important for maintaining blood pH. It's also fully reversible. Now, aside from gas exchange, the blood also plays a key role in moving nutrients, waste, and hormones. Nutrients like glucose, amino acids, and fats, they're absorbed from the digestive system and they're delivered by the blood to tissues throughout the body. Meanwhile, waste products like carbon dioxide are picked up from tissues and they're transported to organs like the kidneys or lungs for elimination. Hormones, which we know are chemical messengers secreted by endocrine glands, they also travel through the bloodstream. They move from their site of release to distant target cells where they can trigger responses by just binding to specific receptors. And all of this happens through the same capillary networks that handle gas exchange. Let's also talk about fluid balance, which is another key part of gas and solute exchange. Inside the capillaries, fluid movement is going to be controlled by two opposing forces: hydrostatic pressure and oncotic pressure. Hydrostatic pressure is the force that blood pushes against the walls of the capillaries. It's highest at the arterial end, and it pushes fluid out into the surrounding tissues. Oncotic pressure, on the other hand, is created by large proteins in the blood, mainly albumin, that stays inside the capillaries and it pulls fluid back in. Albumin is a major plasma protein that's responsible for maintaining oncotic pressure within the capillaries. Now, as blood flows through the capillary, these pressures shift. So at the arterial end, hydrostatic pressure is stronger than oncotic pressure. So fluid moves out. So for example, about 36 millimeters of mercury might be pushing out compared to the 25 millimeters of mercury pulling in. But by the time blood reaches the venual end, hydrostatic pressure has dropped while oncotic pressure stays roughly the same. Now oncotic pressure wins, so fluid is pulled back into the capillary. So you might see something like 25 millimeters of mercury pulling in versus only 15 millimeters of mercury pushing out. And this balance of pushing and pulling is called Starling forces. It helps regulate how much fluid leaves the bloodstream and how much returns. If this process didn't work properly, like if too much fluid stays in the tissue, you can end up with swelling or edema. That's where the lymphatic system really comes in. It's going to pick up the extra fluid and it's going to return it to circulation through vessels that eventually drain into the thoracic duct. Finally, let's wrap up the chapter by discussing coagulation. This is the body's process for forming blood clots to seal injured vessels. When a blood vessel is damaged, the first responders are platelets, also called thrombocytes. These cell fragments detect exposed collagen beneath the vessel lining, and they immediately adhere to the injury site. This contact is going to activate the platelets, and it causes them to change shape and then release chemical messengers. These signals are going to amplify the response, and it will recruit even more platelets in a process called platelet aggregation. Meanwhile, a complex cascade of clotting factors are going to get triggered. And this cascade ends with the conversion of a soluble protein called fibrinogen into fibrin. Fibrin is an insoluble mesh that will weave through the platelet plug. This fibrin mesh stabilizes the clot, it will seal the breach, and it stops blood loss. Once the vessel is healed, the clot is no longer needed. So then the body activates plasmin, which is an enzyme that digests fibrin and dissolves the clot. Plasmin is formed from plasminogen. It's an inactive precursor, which gets incorporated into the clot early on, so it can be activated later during clot resolution. And with that, we've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.